Okay, welcome everybody. This is our 2023 DIPG DMG Medulloblastoma Symposium review. You probably see the screen that you have. We're going to go ahead and get started. We're joined by Daniel Adewumi. He is going to go ahead and he's from Abby's Corner, more importantly. He's also a collaborative partner, and he helped me in trying to get a firm grasp of all the various sessions that we had going on for this symposium. And there was a lot of them. We had about 4 or 5 different sessions going on at once. And so we're going to do our best to just kind of go through each of these individual sessions of note and try to give you as much detail as we possibly can, while also making sure that we can hopefully try to get some of your questions. We may, as you get these, for those of you that are joining us live, you're welcome to put the questions in the chat window. For those of you that are going to be joining this, maybe if we can record this and present this to the general public, just obviously email us. We want to try to help you out as much as we possibly can and give you as much up-to-date information of some really great research trials that we have here. Overall, it was a very big symposium, biggest we've ever had. We had, I think, about 184 people that ended up attending it, and it was very well supported with, my goodness, what was it, Daniel? Probably about 60 or something new research trials, I think, that you have here. You have 74 slides, I know that much, but lots of presentations in the meantime. So without further ado, I'm going to go ahead and turn it over to Daniel to kind of start walking us through this process.
Awesome. Thanks so much, Keith. Hello, everyone. I want to take a brief moment to introduce myself. My name is Daniel Adewumi. I am not a researcher, but I am a collaborative member. Just a bit of background about my own professional experience. I have a bachelor's of science in biochemistry and a bachelor of arts in sociology. Again, I'm not a researcher. I'm currently a JD candidate at Columbia Law School. But again, more importantly, I'm a representative of Abby's Corner. So we are part of the DIPG DMG collaborative as of November 2022, following the passing of my little sister Abby at 16 years old. But essentially, the purpose of this webinar is to give a real compact summation of the research sessions of what was accomplished and discussed at the symposium, such that everyone is able to stay abreast of the current state of research. Awesome. So we're actually going to start with a review of the data collection efforts. So I'm going to turn it over to Keith to kind of discuss some of the registries.
And this is, I can take this because I'm involved in both of the steering committees of both of the registries. The registries are broken into 2 separate groups, one of which being the International Registry, which is comprised of Canada, Australia, the United States, New Zealand. And then there's also the SIAP Registry, which is principally Europe and Mexico. They are both things that we participate in, and we kind of count these numbers a little bit together, but we had 2 separate updates on this. And this really stems from the genesis of in 2015, some of the European privacy laws required that we needed to break these into 2 groups, but they do work together. It's just the databases have to be separate. Within the European registry, that's the SIAP registry, we had 134 patients that are enrolled, which is a fantastic number on its own. We have 634 retrospective, which means that they were collected typically after passing. We have 400 prospective, which means that they are collected while they're in treatment. And there's been numerous studies. They went through a whole list of them. I'm not going to jump into that. But one of the most significant things I think we're going to see out of it is the long-term survivor study. For those of you that may be familiar with some of the history of DIPG DMGs, back in 2017, we had a paper that really is the cornerstone that determines all the statistics that we talk about, the survival numbers, the amount of months, everything that everybody kind of uses as the benchmarks for studies. We have asked through our groups, both Cure Starts Now chapters and the collaborative partners, for an update to that because we feel some of those numbers have changed. We're also working through the European one to increase enrollment by adding DMGs to the registry. We believe that that's probably going to add on about 400 patients to it. Within the international registry, there's 1,113 patients. This also is very much available for scientific study by researchers. We've actually asked that the process is simplified so that that way people can get access to the data a lot quicker. And we have significant amounts of tissue and genomic data. And then we addressed the future directions, which really came down to simplifying the process, working more with pharmaceutical companies. Matter of fact, one was just done here since the last symposium. We had a new one that was added on, and really focusing on getting better at getting new data going forward so we can establish clinical benchmarks. So. I guess I'll go ahead and take this as well. This is the trial finder. We also had an update there talking about a brand new system. Well, not necessarily tremendously new. It's been around for about a year, but it's 2.0. And trial finder is a system that we have collectively as groups developed that allows parents and patients to get every trial that is in anywhere that collects the data, which is Europe, Canada, US, Australia, New Zealand, all trials that are relevant and open or even closed sometimes to DIPG DMG patients. So effectively, instead of relying upon a doctor to remember what trial is available or what has changed and hope that they're reading every single documentation you can find on DIPG DMG, we have a system that actually does this for you automatically and sends updates directly to your phone. So those of you who may already be familiar with it, it can be found at DIPG.org, which is an unbranded site, which is designed to represent the global community of DIPG patients and families and foundations out there. But it's a great way to get trial aggregation without bias, without it being your local hospital, and get it unfiltered. We actually have a few doctors, quite a few of them, that are actually using this to advise their patients. And so the point of this is, let's make sure the patients know it so they get the data real time as well. It also offers some data and patient assistance in it. So there is some free doctor consults that go along with this, particularly for the United States, and even a little bit that we're working on in Australia. This is a great way to also compare, and frankly, the only real way to compare international trials across all the various platforms. And as I said before, it offers some impartial recommendations. You can see the various groups that tie into it and how it queries the sites.
Awesome. Thanks, Keith. So from here, we're going to get into a review of the current research. My goal here is to give compact summations of the research, but in ways that we can digest them, big takeaways from each project. So we're going to go ahead and get started with a study by Chris Jones currently being done, translating mechanism to patient benefit for ACVR1 mutant DIPG. So in this study, essentially, there is a protein called ACVR1. This is a protein in the body generally. It helps, it's found in many tissues of the body, including skeletal muscle and cartilage. It helps with growth and development of bone and muscles. However, there is a mutation in ACVR1 that has been found in a quarter of DIPG patients and was not previously linked to any other type of cancer. More recently, it's been discovered in subgroups of DMG patients as well. So ACVR1 mutations being found in DIPG patients, researchers are examining something called TP0184. This is an inhibitor of ACVR1. Essentially, they're taking this inhibitor, they're utilizing it in DIPG research to see its effects on DIPG. They found that it is inducing apoptosis, which basically means the DIPG cells are dying. It's radiosensitizing DIPG. That means that DIPG cells are becoming more sensitive to radiation therapy. It's also been well tolerated and able to get across the blood-brain barrier. So the blood-brain barrier is something that selects what can cross over past the barrier to the brain. It's a hurdle in DIPG research, as drugs have to be, first of all, effective, but also to get across that barrier. But one thing that they have been finding with this study is that TP0184 has no selectivity. That means it affects both the mutant ACVR1 that we're looking for and also the natural wild type ACVR1 that our bodies produce naturally. However, this drug is being tested on its own and in combination with other drugs. And prior to being ready for clinical trials, it has to go through preclinical guidelines. That means researchers have to determine what is the effect of this drug in combination. Does it have a cellular effect? Does that still kill cells? Does the drug continue to cross the blood-brain barrier? There are an assortment of questions that need to be answered prior to further development. However, there have been encouraging results thus far. So with the next study, this is another Chris Jones study, which essentially focuses on DIPG subclones. So you'll see that in both these studies and the one to follow this, it's examining DIPG cells as being and their heterogeneity. That means the cells can look different, the tumor can look different depending on where you pull the cells from. So DIPG is comprised of multiple coexisting subclones. These subclones are interacting with each other. The whole essence of this study is, can we stop these subclones from communicating? Can we stop these subclones from helping each other move and their processes? So we're looking at which drugs affect these subclones, and we're trying to see where can we do combination therapies? Where can we combine drugs and to stop these interactions? Okay. Study number 3 by Valdez Mora. Epigenetics. Epigenetics concern how cells control gene activity without changing DNA sequence. So that means that epigenetic changes are modifications to DNA that regulate whether a gene is on or off. So deregulation of epigenetics can cause cancer. So in this study, researchers are looking at something called H3K27M. This is a mutation in an epigenetic factor. The takeaway here is that H3K27M is a mutation found in 80% of DIPG cases. But to this point, the mutation itself has not been druggable. So researchers in this study are trying to limit the consequences of this mutation. They're looking at what are called BET inhibitors. They're looking for ways to reduce the ability of DIPG cells to survive in the presence of H3K27M, but they need to find new ways to conduct the therapies, as epigenetic therapies are notoriously tough and rigorous on children. But essentially, again, the takeaway, H3K27M, in 80% of DIPG cases, researchers are trying to limit the consequences of these mutations, as they have not been able to target mutations themselves. Okay, from here, we're going on to the next study by researcher Mill. So again, all of the previous studies have been focused on the heterogeneity of the tumor. So in this study, we're looking at medulloblastoma specifically. So with medulloblastoma, 30% of patients relapse. That means that there's signs of improvement, there's signs of recovery, but there's a return of the disease and the symptoms after a period of improvement. Researchers are trying to examine in this study what is causing this relapse. Is it the nature of the tumor itself affecting the brain environment, or is it the treatment that's administered actually contributing to an environment that encourages relapse? So palbociclib is a drug that has been beneficial in treating medulloblastoma, and researchers are examining, does using palbociclib also create a brain environment where the brain can become stiff and the tumor can relapse? So what is causing the tumor relapse, and what is causing the tumor to relapse and return aggressively? Is it the tumor itself, or is it the treatment that we're administering? So in the next study again, we have researcher Drissi. So this study is dealing with immunotherapy. So immunotherapy is when we try to stimulate the immune system to treat cancer. We're trying to improve on a patient's immune system's ability to naturally fight the disease. Now, when in doing so, T cells are incredibly important to this effort because the T cells in your body attack foreign particles. However, there are a few obstacles in trying to conduct immunotherapy as it concerns DIPG. DIPG is what's called a cold tumor, which means that the surrounding cells around the tumor are able to suppress the immune response. They basically act as barriers to T cells. So this study is trying to see if, through administering drugs like decitabine and nivolumab in combination, can we overcome some of these obstacles presented by DIPG in conducting immunotherapy. There have been studies done with mice. In the mice trials, we've seen that the combination has improved the overall survival of the mice. However, in another model, we did not see the infiltration into the tumor by the drugs that we are looking for. So this is a study that is ongoing, but essentially they're trying to utilize combinations of drugs to overcome some of the obstacles that DIPG presents to immunotherapy. Okay, now we have molecularly guided phase 2 study of ribociclib and everolimus. So this is a trial that we've been with since 2016. a sizable portion of pediatric high-grade gliomas and DIPG tumors have genetic alterations in two key pathways. So ribociclib and everolimus each target different pathways that are overactive in DIPG. So in this study, they're looking at these two drugs in combination to target two key pathways, specifically being alterations in the cell cycle and something called the PI3K and mTOR pathways. So they have conducted a phase 1 study where 19 patients were enrolled, and the 36-month survival rate of these patients was 27%. So it's important to note that with this study, some of the patients who had prolonged survival did not have the cell cycle mutation in the pathways that are under examination here. However, these results do suggest potential benefits of using ribociclib and everolimus in combination. So a phase 2 study is incoming. However, patients need to have the alterations that are under concern here, and tissue collection needs to take place for the patients. But again, the takeaway here is that ribociclib and everolimus each target separate pathways that are overactive in DIPG. Thus, we are examining the benefits of utilizing them together in therapy. So we're moving on to the next study concerning Lutathera. So in this study, we are looking at somatostatin receptors. Somatostatins inhibit proliferation of both normal and tumor cells. This means it stops the cells from continuing to multiply. So Lutathera is a targeted radiotherapy which binds to tumors that have somatostatin receptors. So essentially, Lutathera is trying to locate these tumor cells that have somatostatin receptors to then inhibit the multiplication of these tumor cells. So it's trying to cause cell death through localized release of radiation with minimal side effects. That's the goal of this study. Okay, now we have developing novel combinational therapies for DIPG, researcher Danielle Upton. So in this study, we have a catalog of 3,570 drugs that have been tested. Essentially, the goal is to look for which drugs are effective and which drugs can be used in combination with one another. So one of which was ACT-001. That was one of the most effective drugs tested. It crosses the blood-brain barrier readily. No dose-limiting toxicities have been observed, meaning patients can be administered doses that are required for the therapies without some of the major adverse effects. And in the mice models, it killed the tumor cells and extended survival in the mice models. Now, ACT-001 is being used in combination with CBL-0137. And again, in the mice models, survival was extended. Now, regarding clinical trials, single agent testing must be done before drugs can be tested in combination, meaning that ACT-001 must be tested on its own prior to being tested in combination with CBL-0137 in clinical trials with patients. So it's currently being examined in 15 patients, but the takeaway with this study is ACT-001 and CBL-0137 are 2 of 3 chemicals identified that we saw reactions with. So again, with this large, broad catalog of drugs that are being tested, these are some that we've seen promise with that we want to continue testing for. Now we're going to researcher Cynthia Hawkins, synthetic lethality screening with methionine restriction for DIPG. So in this study, they found that DIPG patient cells, are extremely sensitive to the withdrawal of methionine, which is an essential amino acid that can only be obtained through diet. So they examined in cell lines restricting methionine and found that there is a window where normal cells survive, but DIPG cells do not. So mice in a methionine restricted diet saw improved survival rates, and this was confirmed in multiple labs. Now, a methionine restricted diet looks like a vegetarian or vegan diet. However, I must issue a caution. Sometimes restricting methionine may not be able to be done strictly through diet alone. And in going through DIPG treatment, these types of diets, vegan and vegetarian, may actually create problems during other parts of the treatment process with DIPG and DMG. However, researchers are looking to see the effects of restricting methionine in combination with other types of treatments to see whether or not that can also have positive effects in DIPG research. Now, I'm going to take a second to discuss two Cure Fund grants. So the Cure Fund is something that is an award of $50,000 given to two researchers at the symposium. They're able to apply for the Cure Fund, submit their grant proposals, and then present on these ideas at the symposium, at which point the Medical Advisory Council deliberates and selects two research projects that are worth funding. So the first of which that received funding at this recent symposium is exploring novel targets in DIPG by researcher Dasgupta. So in this study, the goal really is to look at pathways dysregulated in DIPG. So there is a drug, you'll have to bear with me on this drug. There's a drug titled IACS 010759. So the takeaway with this drug is that it is an inhibitor of one of the pathways dysregulated in DIPG. So they're testing this drug's effect on DIPG in mice. And again, this is research that just recently received funding. So it's definitely an ongoing project. Now, the second of these Cure Fund grants to receive funding is Starve the Monster. So this study is essentially looking at metabolic treatments using combinations. So there's a 3-step process prior to analyzing these combinations. But again, there's a theme with some of the research that's coming out of the symposium. We're really starting to look at combinational therapies. How can we utilize different types of therapies, different types of drugs to see where the positive effects to be had are? And this is one of those studies. Now, returning to research that was funded prior, that was presented on at this recent symposium, we have Matthew Dunn, enhancing the recruitment of tumor-infiltrating lymphocytes to improve DMG therapies. Now, this is another study concerned with immunotherapy. So recall, DIPG is a cold tumor, meaning the surrounding cells around DIPG tumors are able to suppress the immune response. But in this study, ONC201 is a drug that has been shown to promote the release of immune cells from bone marrow, which makes the DMGs more visible to the immune system. So ONC201 is meant to help overcome some of the obstacles to immunotherapy in DIPG. It's meant to increase the immune response, and it's being studied in combination with paxilocin together to recruit immune cells to the tumor. The goal here is to increase the oxidative stress, but again, ongoing studies, and we're staying tuned for the results of this. Now we have DMG Combat 1.0 and 2.0. So recall again that 80% of DMGs have the H3K27M mutation. So the takeaway with this study is that paxilocin inhibits pathways observed in 80% of DIPG cases. So this researcher is looking for drugs to use in combination with paxilocin. Enzastaurin could be one of those drugs. However, again, with the blood-brain barrier, which filters what substances are able to make it into the brain, enzastaurin has been found to have poor crossing of the blood-brain barrier. So although enzastaurin has been seen to work well with paxilocin, enzastaurin's failure to cross the blood-brain barrier to the extent that we'd like has caused an obstacle. So this researcher is looking to develop analogs that have the same properties as enzastaurin and the same beneficial effects, but that are able to get through the blood-brain barrier at improved rates and efficacies. Now, that raises a question. Do we develop these analogs and go through the process of getting them through drug approval, which can take some time? Or do we also look to apply new methods of getting drugs across the blood-brain barrier, see if enzastaurin can be made to cross at better rates? So these are some of the questions that are arising with this study. However, essentially, the researcher is looking to use paxilocin in combination with enzastaurin or drugs of similar properties. Now we have researcher Dasgupta again, targeting purine synthesis and 1-carbon metabolism in DIPG. So the takeaway with this study is that researchers discovered 2 key genes in the metabolic pathways that make the building blocks for DNA and RNA that are novel targets for DNA. So 2 key genes. DIPG has high levels of the enzyme ATIC, ATIC. So in this study, in the mouse models, targeting ATIC has led to a clear survival advantage. Essentially, how does that continue outside of the mice models? In continued development, what is the effect of targeting ATIC on DIPG? Now, for this next one, I'm going to actually pass it over to Keith to discuss AMXT 1501 with researcher David Ziegler.
I think this is a well-placed time for you to get a breath and maybe a drink a little bit. Thank you, Daniel. Daniel's done a great job, as you guys can see, because it took me 13 minutes to get through 3 slides. He just went through 32 slides in 13 minutes. So obviously, he does a really good job of summarizing it. We're very appreciative to have his help with this one. I'm going to take this one because this is one I'm working on. This is AMXT 1501, and we've been familiar with it since 2011, and the DFMO combination. This is really focusing on these polyamine pathways and using this as effectively the Achilles heel to be able to focus on with this tumor. We find that DFMO tends to work well. The good news is this is not a single agent trial. We're trying to take DFMO and AMXT and try to put it together. It does use an IV pump that's called a CAD pump. It is a pump that they have to be hooked up to for the entire duration of the treatment process. That gets to be a little bit of a hurdle in some of that. But this is one that just advanced to a clinical trial. What you're seeing is what we funded on the translational model, and we showed that 2/3 of mice had tumor regression. That's a very amazing number, one of the best numbers we've ever seen in DIPG DMG. And so this advanced rather quickly for clinical trials, and it has even gone to a compassionate use request to try to get it so that it can go to patients less than age 12, because the FDA kind of keeps us in those parameters. Certainly a very interesting trial, one of the most positive we've seen thus far, but that's why you do trials, is because you just don't know. But certainly one we're keeping an eye on very much within this, and we'll continue to support as we go down the road here, just because we're seeing some very interesting things that we like. I'm going to go ahead and hand it back over to you, Daniel. You ready to go?
Awesome, let's do it. So I didn't realize it had only been 13 minutes. I hope I'm not going quite too fast, but I'll try to continue to make sure we get the major takeaways out of these studies. So with this study, researcher Eric Thompson identifying oncolytic viral therapy resistance mechanisms in brain tumors. So the essence of this study is to look at what's called viral immunotherapy, which is the use of a virus to stimulate the body's immune system. This could function as a replacement for toxic chemoradiotherapy. So we're trying to, with this study, look at the immune response or activation in response to viral therapies. We want to see how the body responds to viral immunotherapy, and can this response or reaction be controlled? Now, our next study is researcher Ian Pollock, the role of QPRT and NAD pathways in DIPG treatment resistance. Now, this is one of the more complex studies, so I'll try and break it down to the major takeaways here. We have what are called proteasome inhibitors and HDAC inhibitors, which both typically assist the body in fighting off cancer cells. So panabinostat is a drug that is a HDAC inhibitor that crosses the blood-brain barrier. Meanwhile, marizumab is a proteasome inhibitor. So we're trying to utilize these two drugs in combination. Now, in looking at these two being used in combination with mice, we've seen that it's killed off 80 to 90% of relevant cells. And in addition, the cell killing affects, it's killed off 80 to 90% of relevant cells. However, the cell killing effect has been reduced due to resistance buildup. So recall that with this tumor, as it undergoes treatment and therapies, it can kind of build up a resistance to the therapy that's been effective thus far. So these two drugs in combination have been effective, but the tumor has been building up a resistance. So one way of trying to reduce this treatment resistance is by targeting what's called glycolysis, essentially trying to starve the tumor. And one drug that is useful for that is lenalidomide. So in using lenalidomide in combination with panabinostat, we've seen, again, an increased survival rate in mice, both in mice that have built up resistance to the tumor and in mice that have not. So this is also somewhere where ketogenic diets have been examined in combination with some of these treatments. The results with that were a bit less exciting. However, the essence of this study is panobinostat, an HDAC inhibitor, marizumab, a proteasome inhibitor, and then the addition of linidamine to keep the tumor from building up that resistance. Next, we have Richard Liu targeting histone lysine demethylases to treat DIPG. And again, we see the return of the H3K27M mutation. Now, remember prior, I said that to this point, H3K27M as a mutation has not been druggable. Research regarding this mutation has been trying to limit the consequences that take place when the mutation is found. However, this study is seeking to target the mutation itself rather than simply limiting its effects. So an epigenetic screen identified something called chitosan as a new target in DIPG DMG. So treating chitosan treatment has decreased proliferation of H3K27M DIPG cells, meaning they have not been able to multiply and reproduce at the same rates. It's also selectively increased apoptosis in DIPG cells, in DIPG lines, and has prolonged survival in H3K27M DIPG. So essentially, chitosan treatment has decreased the ability of these cells to multiply. It's selectively encouraged these cells to die and has prolonged survival. It is a fairly recently funded study as a part of last year's cycle, and much of the research is ongoing here. But again, the takeaways have been quite encouraging thus far. It's searching to target the mutation itself rather than just limiting the consequences. Now we have viral immunotherapy for pediatric brain tumors, Candelaria Gomez-Manzana. So this is looking at viral treatment using a virus that has been changed in a laboratory to find and destroy cancer cells without harming healthy cells. So researchers have found evidence suggesting that viral treatment induces an anti-tumoral immunoresponse. So treatment using these tumors is encouraging the body's immune system to take the fight itself. So we have seen reductions in tumor area when this method has been tested. But remember, this tumor often builds up resistance to treatment as it's ongoing. So overcoming resistance isn't something that this study has been able to examine yet, as again, this is another new study that is ongoing. So the first few trials, the theme there was the way that this tumor and these tumors, the heterogeneity within them. How can the tumors look different? How can they develop differently? Then we got into some of the treatment methods. We're looking into ways to combat this tumor. Now we're getting into an area where we're looking at therapeutic delivery. How can we actually administer therapies in ways that are more effective in combating this illness? The first of which is intranasal delivery of targeting nanotherapeutics for DIPG. So we have what's called convection enhanced delivery. So when we're trying to administer drugs to something like DIPG in the location that it is, it's difficult to reach, passing the blood-brain barrier, administering drugs to high enough doses without negative effects for patients. So convection enhanced delivery is a method of drug delivery in which the drug is administered using bulk flow rather than conventional diffusion into the brain. So it's a very effective but very invasive method of delivery, as it needs a catheter inserted into the brainstem. So in this research, the researcher is looking at intranasal delivery through the nasal cavity. So it's much less invasive. It has rapid delivery to the brain, bypassing the blood-brain barrier. It's less effective than convection-enhanced delivery and than IV, but it's much less invasive as well. Takeaway here, we're trying to find another way to get to the tumor. Now we have phase 1 of 2 study of sonodynamic therapy using Sonolad 001 and Exabolate 4000 type 2, researcher Stuart Marcus. So this is another study about how we can get to the tumor. This is metabolic targeting of the heme pathway. So ALA photodynamic therapy has been seen to selectively kill tumors, meaning targeting tumor cells while maintaining the health of normal cells. So it's been used at 2 clinical sites thus far with positive results, but it's not yet commercially viable. However, they saw significant tumor regression and extended survival. The median survival was actually 39.7 months. So this is a study that we're continuing to follow. It's sonodynamic therapy. Sonodynamic therapy causes tumor regression by the photodynamic effect. This means that when you look at the tumor, it has a cell membrane around the cells that are kind of the barrier as it relates to the environment and to other cells. It's trying to disrupt the membrane, basically break the membrane, allow for the cells to be killed in that way. So regarding the safety data thus far, there have been no acute or dose-limiting toxicities observed in the 4 patients treated, meaning that patients are able to receive the dosage and the amount of administration necessary. And there have also been no negative lifestyle changes outside of hair removal. So again, this is a study that we're following. This is looking for a way to get to the tumor and using photodynamic and sonodynamic therapy to try and attack the tumor. Now I want to pass it over to Keith again to discuss another Richard Liu study, FLASH, essentially.
Yeah, and this has a different title, and we're making another transition. I mean, Daniel described how we have this changing in our strategies. That's really what I focus on more than anything else. And this is another one of those. And this is really looking at what things we can do with radiation that may provide better assistance in the future and possibly even give us almost a reset button so that we can make changes in between trials, realizing that perhaps we perceived radiation wrong and it could be doing a little bit more with some improvement. And we talk about it as FLASH, and this is a pretty simple one to talk about because while this trial has a very complex title and maybe focusing on different types of drug deliveries, what we really see in this is we see the development of this thing called FLASH. And what FLASH is an ability to be able to deliver a high dose of radiation through proton therapy and be able to do so in a very short time frame. And we've seen some results that come from some bone cancers where FLASH has been able to work. And so our question dating back to 2019 has been, well, what could that do for brain cancers? And so we've been asking for this for quite a while, and it's something that is coming out through this trial in particular. The hope is that this will allow us to be able to use in between trials to allow a high amount of radiation to be able to impact it only on the tissues with very little ancillary damage, kind of like what we're seeing in some of the bone cancers right now with FLASH, and be able to do it in maybe 3 days or less. And boy, if we can find a way to be able to do that with very little ancillary damage, I think it'll be very impactful going forward to change how we do radiation as a whole. So that's the hope. This is a long way to go. Of particular note, and one thing I forgot to put down here in the bullet points is that this is being watched very carefully by organizations and consortiums such as PBTC, the Pediatric Brain Tumor Consortium, which is the NIH-sponsored arm of effectively pediatric cancer research funding. So it's certainly got a lot of awareness, and it's being based also in Cincinnati because Cincinnati has one of the few, if not maybe the only, research gantries for proton radiation out there. And so this allows us to really test this out in ways we can't test in other places. We're going to continue this moving forward with one of the other trials that's also talking about radiation and other ways of being able to deal with it. So I'll let Daniel take this one.
Yes, and this is actually the final research study that we're going to discuss. From this point, we're going to allow Keith to discuss some of the therapeutic showcase and some other highlights from the symposium. But this study is by researcher Van Zanten, Targeted Radiopharmaceutical Therapies for Pediatric Brain Tumors, Target First. So in this study, this is a study that's been started recently concerning targeted radiopharmaceutical therapy. It's a different way of radiating so that we can do it multiple times. So typically, in traditional external beam radiation therapy, radiation is produced through a machine and sent through the body. So the radiation gets to the tumor, but it also hits everything between the machine and the tumor. The goal of this research is to administer radiation systematically through an injection of a radiopharmaceutical. So you're injecting a radiopharmaceutical composed of radioactive material, which then gives radiation off to its environment. So rather than radiating everything between the machine and the body, the goal is to have a much more directed administration of the radiation such that there is more preservation outside of the tumor and ability to radiate multiple times. But that actually is the crux of all of the research sessions that we had at the symposium. But I want to give Keith the opportunity to also discuss some of the therapeutic showcase that we were able to take part in.
And this was a new thing that we had never, well, we've tried it in the past, and the whole purpose of which was to try to get not only just foundations and researchers together, which was kind of the highlights of what made the symposium special dating back to 2011, but also it was to focus on trying to get maybe the industry and pharma groups to be there as well. I'm not going to concentrate tremendously on the individual companies with it, but it was the biggest group that we had ever seen. Starting out with PTC Therapeutics, and they wanted to obviously talk about their PTC 596 compound that's being utilized through some trials through the Connect consortium. We then had some discussions from Chimerix, and Dr. Nazarian had talked about the role of ONC201 and some of the, maybe an ONC206, but also some of the other permutations that are occurring there with it and some of the results that they saw with it. Kazia was talking about through Dr. Dunn, Paxilosib, chemical and how, or compound and how that's working in those. Bexian also introduced their compound and some of the implications that it had with DIPG and DMGs. And then Insight Techs talked about effectively some of their different ways of attacking it and kind of providing some aid efforts that help to make things more effective in some of the treatments. We had Emulet that was also talking about something similar in that realm, but they were talking about an ability to be able to have a device that is wearable that may allow us to be able to have increased permeability. And then Aminex was talking about the AMXT compound, particularly that DFMO trial we talked about before. Big takeaway with therapeutic showcases, this has never been done before. The thinking is to try to get them in the same room, to give them their 7 minutes to be able to quickly talk about the strengths, the weaknesses, and how this may work with existing trials. And the hope being that we can form brand new clinical trials from researchers who are going to take some of these compounds, hopefully be able to get access to them free of charge for the patients, and be able to deliver a whole new onslaught of clinical trials. The results are still out. We're waiting for some of our biggest grant cycle we expect ever, which should be in here within the next 2 weeks. But already the information that we're hearing is that this was successful, and we should see some clinical trials because we need them. I mean, we got a lot of going on in the translational side of things. We just need to get something so it becomes more bedside. That then concluded our research presentations and what we had talked about literally in a day and a half. So that's a lot to cover in a very quick time frame. There's a couple of ancillary sessions that we did want to discuss with you, though, just to make sure that you knew that they were ongoing as well. And so that's what I'm going to cover in the next section of this. One of these is registry. We talked a little bit about what the registries are, how they're comprised, but what we did is we wanted to have a forum directly with the patients and directly with the families to talk about how it's set up and how we can improve upon it. And so we reviewed the CRF setup system, and what CRFs are is effectively panels or categories of data that we collect. If you go through and you count up, for example, the International Registry, you're going to find out that there's probably almost 700 different collection fields, if you will. But of those different collection fields, one of them may be their entire radiological history. So some of these fields could be huge and could have hundreds of thousands, if not even 1 million data sets in each one of them. And so it's a massive, massive system that we have. And what we did is we went through these individual fields to try to get some ideas of what aren't we collecting. And then we had an open discussion of how we do that data collection and how we maybe get that a little bit more forward thinking. There was also some discussions about some family additions, and do we start to open it up beyond that? And matter of fact, even at the actual symposium itself, we had some data collection that was being done through familial relationships, parents, to be able to help round out some of our understanding of where this comes from and hopefully some of the origins of it so we can stop it. We had something that people have been asking for a long time, and we were able to actually be able to pull off this time. And that's some discussions of dietary and lifestyle changes. And this really came about through 3 separate sessions that were orchestrated by Grace and kind of trying to pull this together. But she got Dr. Adrienne Scheck and Samantha Chacon to be able to discuss it both from a doctor's perspective and start to go through some of the actual evidence that we have that maybe there's something more to this than just kind of this ancillary discussion that always occurs on the sides of these conferences. And then we had Samantha Chacon, who then went through how this actually applies. What are some of the things that patients may be able to do as well? And this is a very complex topic to kind of dig into. And I also want to reference some of the caution points that Daniel had brought up earlier on, in that this isn't for everybody. And it does change as long as you go through the pathway process on it. But what we wanted to do was to at least start that discussion. And so on the medical side of it, we talked about the ketogenic diet and how it may help with some of the inflammation, angiogenesis, peritumoral edema, hypoxia, expression of various genes, anti-tumor response, the efficacy of the P13K inhibitors, and some of the microbiome. But at the end of the day, the question is to what relation this has a lot with DIPG, DMG. And we didn't have a whole lot of evidence that really kind of tied it directly to that. We had a lot of things that may have tied it to some of the brain tumors and to some of the other cancer things. And so the hope is, and we're discussing with actually Dr. Scheck with right now, is what can we do to try to customize that plan so that maybe it can be tied into some of these trials that we're going to be doing as well. On the patient side of things, we talked about how this ketosis as a metabolic state was more of a metabolic state than even as a diet. And really some of the practical things that you can deal with from the high fat, low carb, normal protein, low protein, how fasting impacts that, some of the, in combination with that time restricted eating, caloric restriction, and how to mimic some of these diets. Not going to dig tremendously into this because once again, this by itself is another 2 hours. But I know this is something that a lot of you have actually asked for, and we were able to get together a panel to be able to pull this together. So hopefully we can take this past just a question and actually into a set of recommendations here shortly. We then had a discussion that was set up for dealing with siblings and parents and how we deal with some of these diagnoses. And this was done by Emily McNulty of PTC Therapeutics. They already had a great system that they were delivering out to the family communities, and we asked them to partake in that to help us with this too. And so they looked at some of the developmental considerations of this. They looked at how parents and siblings dealt with this. And then we also had a panel from Melissa Fleming from a parent's perspective, and from Grace Desserick from a sibling perspective, to be able to give us an idea of really what it feels like behind the scenes for those of us that may not been on that other side. And then we had a practical setup of what really should we be doing, what should we be considering as we move forward, and where to go to get more resources on that. We also had a continuation of something that we started in 2017, and that's Club 22. This is done really in tribute and in inspiration to the efforts of Lauren Hill and also Brennan Kelly, two young adult fighters of DIPG. Sadly, they're not with us anymore, but both of them really took up this campaign to try to get adults and young adults to be able to talk about it. And for those of you that weren't around in 2017, this was something that the adults and the young adults didn't want to talk about, because when they would come out, people would tell them that it may have been misdiagnosed or they had it wrong. And so it really took some years to try to get everybody to be out there and talking. Alexa Huber, a Club 22 member herself, helped to lead this in combination with Grace, who's coordinating this. And they put together a really strong panel of folks to deliver some recommendations that we want to see out of the research community and also out of the foundational side of things. They listed them up and were very clear with these, and these were wonderful. One of which is they wanted to eliminate age restrictions on trials. And this is something we've already started doing since 2019. We hope to advance this and do it really system-wide for everybody. Number two, we wanted to get a better correlation of adult and pediatric tumors and get the experts from adult tumors to come in that same room. And that's been a challenge. But the hope is that with the Club 22 support, with the entire community support, we can try to get these folks talking together so we can learn from each other. They wanted better biopsy messages. We wanted to see whether or not should be done, shouldn't be done. I think in the pediatric community, we have that pretty well taken care of. We want to get the biopsy messaging with adults done a lot better. We want to do better at collecting atypical data. I think many times we tend to pass off atypical data as it's misdiagnosed. And sometimes that's the case, but other times it's not. And in particular, when we get into adults, we do want to grab that a lot better because perhaps that atypical data is just really a tumor that is advancing or has maybe endured longer. And so we want to look at that and maybe consider that in the time and space calculation of how we deal with those tumors. The last thing was talking about reirradiation in adults, which is something that's really not talked about tremendously well in the adult community. And that has to change. And I think that's going to be certainly helped by the number 2 point, which is to really get those adults experts into the same category or in the same room effectively is what we're doing. So the hope is going to 2025 that has changed and we're able to accomplish that a lot better. One of the overriding themes that was happening really in these sessions, a symposium isn't just about what we know or what we're reporting upon. It's about questions that we don't know and things that we want to do better. And one of which is dealing with the multimodality and combination discussion. This was been really pushed by Dr. Adam Green and Dr. Mark Swadan. And they've done a really good job of doing this, but this has been something we've been working on for about 2 years, 3 years thus far. And this was us trying to get a group together so that we can advance it. And this has been occurring a little bit on the side as well. So this isn't just a start of it. This is just a continuation of it. But we had in the doctor session a good 4-hour process where we brought together the best experts in the world to come together and to talk about how we can look at some of these trials in combination with delivery or some of these trials with other types of compounds. And really look at this not just from a single agent, multi-agent system, but also look at it from how does it interact with the delivery methods? How does it interact with the dietary concerns? How does it react with the radiation? And how do we make these trials without exhausting the accruals? And the accruals is obviously the amount of patients that are out there, and really do this in a formative way that delivers this in the best interest of the patients and to really put their concerns foremost and get there faster. So this, I can't tell you there's necessarily a conclusion to this. This is an ongoing discussion, but I think what we noticed that there were certainly some leaders in this field and leaders that I think we want to get into smaller group forums to really build this up. And that's where we talk about the task group that we're trying to pull together. For the first time, we also had a sideline of a medulloblastoma workshop. You may have seen this kind of be in some of the trials that we have, and that's because some of the research has been done in medulloblastoma that we found that has been very helpful in the DIPG DMG realm, and also a little bit of the opposite. And so with that, we kind of looked at it and said, we should probably start looking at some of this, particularly as we're looking at the heterogeneity of the DIPG tumors. And there's more things that are maybe more common, not to all types of medulloblastoma, but to certain types. And so we convened a special group of Dr. Roger Packer, Dr. Nick Guittardo, Dr. Margajar, and Dr. Till Mildy to come together and to really develop the starter, the foundation of maybe what will either be part of this in the future, or it could be a separate session. It really just comes down to how much they tie in together. And we want to make sure that we're not wasting the time, valuable time that we have for these symposiums, because as you can see, there's a lot packed into a symposium, and we want to make sure that we accomplish it. And for this situation, we had 5 different sessions ongoing at once, so it's just a lot to take in, and hopefully we can build upon it. Specifically to medulloblastoma, we dealt with risk stratifications, and this is effectively looking at, are we dealing with 4 different types, or are we dealing with 13 different types? And there's some back and forth on this right now with some of the WHO reports out there. We then also had to establish a base of what do we have going on out there within COG and what's really our trendings that we're going to see. So hopefully, as we start to layer on some of these trials, we can see how they fit together and really develop a puzzle piece here. We also want to look at really the folks that have really advanced this, and that being St. Jude and also SIOP-E. This alone was a 4 or 5 hour session. So I'm really oversimplifying it here. But I think what it did is it set the framework that we can really build upon it and really start to develop something that ties in long term. That effectively is the symposium. For those of you that attend it, you know that it's very tiring. It's all involved. We run from 7 o'clock all the way up until 11 o'clock at night. And these 3 days, you just feel like you can't grasp it all. Hopefully we did a really good job. And I'm thankful for Daniel, your help. I don't know if you're still there. Yep. You haven't fallen asleep yet. With your help of really trying to provide a condensed summary of this, there's a lot here though. And certainly we welcome your questions. We'll try to answer them as best as we can. You can throw them in the chat while I tap dance a little bit. But even beyond that, if you're seeing this and it's recorded, just shoot us an email. You can always email us. My email is very simple. It's keith@thecurestartsnow.org. And if I can't answer the question, we'll try to tag you into whoever we think can help you out with it. The symposium is just getting bigger. It's getting more involved. Normally we had our funded research and we covered it in 1 day. We had to go to a day and a half this time. And we even had to start us, an hour earlier in the morning and run an hour later in the evening just to try to be able to cover it. We believe it's going to get even bigger going forward with that. And as we move into 2025, which is our next symposium, we got a lot of homework to do. And our next symposium is going to be in Alberta, Canada. You can always see the updates on it at DIPG.org. That's how all the collaborative members, how all of our chapters, we all work together in that one place so that it's just about the families and just about advancing research.
Hey, Keith, there is a question in the chat.
I see it. We have a question about flash trials and radiation in Cincinnati and how they're beginning with adults and moving to pediatrics. Are they looking to begin trials across ages? It's really in its infancy. I mean, honestly, I don't even think this has necessarily been approved in bone cancers yet. And so we're still at the mouse model levels with it. And we really got to show that proof of concept and that safety efficacy before we can take it to that next level. I hope to get an update. And for this one, I'm probably going to be your point of contact on it. But I hope to get an update next week. We're actually convening some of the folks that are responsible for the trials to talk about what we found out and where we go for next steps on it. But at this point, it's a little bit of a dream. It's a little bit about seeing some really neat stuff that's happening in other types of cancers and saying, well, wait a minute, why aren't we trying it here? And that's where we're at. So I don't want to get everybody too excited about it, but I would tell you this is probably one of the 4 things that I'm watching for. But I don't think this is going to be at really a fruition level for our community, probably for another 2, maybe 3 years, unfortunately. Daniel, with that summary, while we're seeing if we have any other questions, is there anything that you can think of to add? I mean, what was your impression of the symposium, having attended the first one and grasping it as much as you did too?
Yeah, I think it was definitely an impactful experience. This was my first symposium. I think I tried to prepare myself for just the breadth of information that would come in and in and in, but there's really no preparing for it. There's no predicting how it's going to be until you're there. But I really enjoyed the experience. It was helpful for me to not even just from a community level, having people of similar experiences, common goal, being unified on that front, but also to see the progression of research. Where were we 2, 4, 6 years ago? Where are we now? What are we studying? What are we projecting? It really also helped to contextualize the efforts that we're putting forth and the purpose of our communities, our collaborative, our respective organizations, where the dollar's going to, what are the beneficial results, and how can we continue to move forward? So I really enjoyed it.
Yeah, and I would totally echo that. I mean, you got 70 plus chapters and foundations that are working together and basically putting all credit aside, and we all just get together and we say, what do we need to do next? How do we do it better? And the families, I mean, and I'm not even counting the warrior families. My goodness, we have 800 families that are joining us in this effort too. So this is the biggest collaboration effort in DIPG DMG, bar none. And it's having some really, really good results to it. And the question we get asked by a lot of people is, how close are we? And, as a parent, having gone through this with my daughter and, 17 years ago, I think we're closer than we've ever been. I mean, because that's one of the answers, but that's not really an answer. I would tell you that curing this type of cancer is kind of like putting together a puzzle upside down. You can't see the picture. You don't know what it's going to look like, but you know some key things that you can do that you can improve your process. And so what you do is you go through and you start looking for the edges and you build your edges around the outside. And now at this point, I think we have our edges built. I mean, I can tell you, I see serious progress in this and I see a frame now. And so now for the first time, the challenge is getting researchers to really come together and to find where they have matched pieces, how they're trying to build that. And so now I think it's really just a little bit of the tedious work of just effectively making one puzzle bigger and building it out till we start to get everything formed in so that we finally know what this actually looks like. And that's going to either go fast by everybody working together and doing it in a transparent manner, or it's going to work slow. But hopefully these types of events really bring people together in ways that they just don't ever do. I mean, I have one thing for those of you that attended it, you may see, and that's this puzzle. And this was my running theme throughout the entire thing. Dan, you remember doing these. How many puzzles did you do that night?
I've got mine on the counter, actually. I'm about to go grab it now, but definitely there was a big push for these puzzles.
Yeah, and we did this, it was a little bit tongue in cheek, but the whole idea is we wanted people working together. We wanted to help get them to talk. And so they're sitting at a table, they don't know the person next to you. And so while they're sitting there having dinner, we gave them an assignment and we put broken puzzles, all the pieces laying in front of them and said, now work with each other and try to put the puzzles together. And then we went around and tried to work with them. That's what we're doing here. We're trying to get a community that hasn't really talked to each other because there hasn't been consensus. There hasn't been an excuse to focus on it. DIPG researchers were very few in nature and super smart. They just didn't know how they connected. And so what we're trying to do by virtue of these symposiums is get them to connect and solve the puzzle as it were. So I think that covers us. Any final words? Okay, I think we got a summary in 1 hour. Like I said, if you guys have any questions, we'll put it up on the final screen here, an email address. You can send us an email. We'll try to answer as best as possible, but we thank you for joining us here. And once again, thank you for being part of the community and helping to get all this cured. Have a nice afternoon or nice evening, nice morning. Goodbye.